welcome to week 3 of Biology 212. Today we're going to talk about mutations, and to talk about it we're going to kind of make a flowchart together. So get yourself a pen and a piece of paper and get ready to write down some notes. Alright, so I have my piece of paper, I've got my pen, and today we're going to think about mutations. Mutations. How exciting! So the first thing we need to know is, what are mutations? Mutations are going to be permanent changes. Permanent meaning it's there forever. Changes in a DNA sequence. So permanent changes in a DNA sequence. And something else we need to know about mutations is that they're not always bad. Like when I think about mutations, I think about like Spider-Man getting bit by the mutant spider and developing powers, and you know, that would be cool, so that would be an example of something good. But that's not actually how mutations work. Instead, these changes in the DNA may or may not affect how your body produces proteins. And if you get a change in the DNA that has no difference in how you produce proteins, then it's not a bad thing. But sometimes you get a change in a protein that makes it more likely to have your cell develop cancer, and then it's a bad thing. Most of our mutations are kind of neither good nor bad. It's just situationally dependent. So we know what they are. We know that they're not always a bad thing, so let's not judge our mutations. They happen to all of us. We are all mutants. It's lovely. And then the next thing that we need to know is how we get mutations. How do you get a mutation? Turns out there's 2 ways you can get a mutation. One way that you can get a mutation is it can be hereditary. When something is hereditary, that means you inherit it from your parents. If you're inheriting it from your parents, that means that it was in your parents' sperm and egg. And so hereditary mutations are often also called germline mutations. And they're called germline mutations because they're present in the sperm and egg. Now, if the sperm has a mutation and it fuses with an egg, creating a zygote, that zygote will do mitosis and grow into a person. That means that whatever mutation was in the sperm is going to be in all cells of your offspring. Offspring being your kid or your baby, not just a great 90s band, highly recommended. So that's what makes this germline mutation so interesting, is if you have it in your sperm and your egg, if you have a kid, your kid's going to have it in all of their cells. Another way that we can get a mutation is to acquire it. We call these acquired mutations. An acquired mutation means that you are not born with it. It occurs after birth. So this could be something that is only in some of your cells. Since you weren't born with it, it's not always in all of your cells. And a lot of our acquired mutations are environmentally caused. What do I mean by environmentally caused? That would be things like toxins in the air, or your water, or your food, pollution, UV radiation, too much exposure to sun. This could also be environmental, like what you're choosing to do with your body, like if you're a smoker, or if you have exposure to the HPV virus, that causes mutations in cells. So these are environmental. They happen after you're born. You're not born with them, and it will occur in some cells. So like if I have HPV, the cells that are going to be mutated would be my cervical cells. If I were going to smoke, the cells that would be mutated would be my lungs and my upper respiratory tract. So I'm affecting some cells, but not all. Cool. So our mutations, changes in the DNA, now we know how we get them. Let's talk about the types of mutations. I'm going to keep my list going because I'm into this organization right now. Types of mutations. We have three main types. We have mutations that have no effect on the protein you're making. So this would not be a bad mutation. This is a harmless mutation. We call those silent. A silent mutation does not change the protein. Let's take a moment and break this down. So we know, this is just like our thought bubble now, okay? This is our side note to ourselves. We know that DNA is so important because DNA has these little pieces called genes, and genes are used to make proteins. So if I'm going to mutate my DNA, if I mess up a gene, that means I might mess up the protein. And this could have really big effects. If I mutated the DNA in the region of the gene that carried oxygen in my blood cells, that means that the protein in my blood cells that carries oxygen could be defective, and I could have a different form of anemia. I'm not getting oxygen to my tissues. What this looks like on our mutation level is if I have a genetic code that's T, 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 C, okay, I've got this little piece of code. You know what the other complementary pair is. T always binds with what? A. So A, and what does C bind with? G. Fabulous. So if I have this code here of DNA, and I'm only looking at one side of my DNA double helix, and I want to figure out how to make a protein from this, I'm going to read my code in 3 pieces. Those 3 pieces that we read together are called codons. And you're going to learn a lot more about this in the next chapter. This is just a brief introduction so we can explain how a silent mutation does not change a protein. Each set of 3 nucleotides is called a codon, and each codon codes, that's how I remember it, for an amino acid. But it turns out there's some redundancy. So TTC, this amino acid is called lysine. That's what this codon puts into the protein, because proteins are made of amino acids. The monomers of proteins are amino acids. So if I were to make a little bit of a change to this codon, let's say I changed the C and I made it a T, so that's my mutation. I've changed the DNA. It turns out that TTT also codes for lysine. So I have totally mutated the DNA. This C is no longer there. It's now a thymine. but I haven't changed the amino acid, and that means that I won't change the overall protein. So this mutation is silent. It did not change the protein. I'm going to set this up. We're still working on types of mutations. We have one silent. Just going to do this again. One silent. We know a silent mutation does not change your amino acid. Our second type of mutation is going to be called a missense mutation. So it's no longer making sense, it's missense, and this is going to switch around one amino acid. What does this look like? So if I use my original example where I had the codon TTC and TTC coded for lysine, let's say I change the second T this time into a C. So here's my mutation. That no longer codes for lysine. Instead, this codes for arginine. So I have changed my amino acid. That means that my protein has changed. It may or may not function the same way. Maybe that change didn't really do a lot to my protein. Maybe it made my protein more active or less active. Who knows? It totally depends on the type of protein. But I have changed the amino acid. It is a missense. The third type of mutation is a nonsense. There's no sense happening in this mutation. And what this mutation does is it inserts a stop. Inserts a stop signal. That means that if I had a protein that was, let's say, 20 amino acids long, and I had a mutation that changed an amino or a nucleotide and inserted a stop signal, it would truncate it. Truncate means to cut it short. So if it should be 20 amino acids long, each one of these little circles represents an amino acid. So here are my monomers. I'm tying them together with peptide bonds. This is going to be my protein, so this is now a polymer. This is my protein. If I had a mutation right here between my third and my, what is that, eighth amino acid, I would break the protein here and it would have this short little bit, and I would never grow the second half of my protein, and my protein would be broken, essentially. Most proteins that are truncated like this are no longer functional. So it could ruin it. This is the most serious of the types of mutations. Silent does nothing really, because your protein's still the same. Missense changes one amino acid, which may or may not have a big effect. Nonsense inserts a stop, and that has a huge effect. It could totally destroy your protein. So those were my types of mutations. Let's talk about how they occur next. So keeping my little pattern going on. Mutations occur in 4 ways. The first thing that can happen is an insertion or a deletion. An insertion is adding more genetic information. A deletion is removing genetic information. And this could be adding or removing. It could be a nucleotide. It could be a gene, or it could be a whole piece of a chromosome. So like that's pretty intensive. We'll talk about that again in a minute. So I'm going to add something, a nucleotide, a piece of a gene, or a whole chromosome, and that's going to change my DNA sequence. Where I didn't have any before, I have more, and where I had more, I now have less. The second type of way we can mutate our DNA is through a duplication. A duplication is taking a piece and repeating it. So, repeating a small piece of it. Then, to get a little bit more intensive, we could have a repeat expansion. So this was repeating a small bit. This is going to be a chunk of DNA. Could be several nucleotides, could be a couple hundred nucleotides, that gets repeated several times. So not just once, not just a little piece, this is a large chunk, and it could be several repeats. So repeat expansion, that's telling you a big part of it is happening. It could be repeated 3 or 4, even more times inside of your genome. Our third way a mutation can occur is called a frameshift. Sorry, fourth. So a frameshift mutation is really interesting. When a frameshift mutation occurs, it's either the addition or the loss of nucleotides or the duplication of nucleotides. So either of these 3 ways can cause a frameshift mutation. And what happens is it ruins the whole stream and the whole pattern of your DNA. I say ruins. It doesn't have to be bad, but it changes it. So maybe we should get rid of ruins, changes. So I'm not perpetuating a negative stereotype about frameshifts. It changes the whole DNA sequence from the mutation on. So that's kind of the key, is that it goes further than just where the mutation is. Let me give you an example of this. So if we pretend that this sentence here is a sequence of DNA and we are going to create a frameshift mutation, this is how it works. So I'm going to make a sentence with 3-letter words. The fat cat ate the rat. Look, it's a sentence. That's great. I'm using 3-letter words because I want to represent codons. So each of these codons would code for 1 amino acid. If I had a frameshift mutation, I would be doing a insertion or a deletion, and it would change the whole scheme of my sentence from the mutation downward. So for example, the fat, and then I'm going to insert a nucleotide. So this is my mutation right here. I've inserted a nucleotide. That pushes the rest of my sentence down. The rest of my sentence ends up looking like this. Fat cat ate the rat. So you can't even tell what that sentence used to be anymore, because this insertion pushed the rest of the information down, and every single one of my codons downstream from the mutation got messed up. So now this amino acid is no longer the same, this amino acid is no longer the same, and it shifts the entire protein into something totally different. That's why it's called a frame shift, is the entire reading frame downstream from your mutation gets messed with. Woo, pretty neat, right? Frameshift mutation. So the last thing I want to talk about as we talk about mutations is what happens if you do that chromosome change that we kind of alluded to in our first way we can have a mutation occur. That's going to be called, make sure you can see it, yeah, aneuploidy. Ooh, cool vocab word. That's a change. Oops. In the number of chromosomes. A change in the number of chromosomes. Humans usually have 2 sets of chromosomes. We call that diploid. We have 2 sets of chromosomes in each of our cells. But there's totally differences between people, and some people have more than 2 or less than 2. So if you have more than 2 sets or less than 2 sets, it's an aneuploidy, and then we can get more descriptive. So if you have a trisomy, that means that you have 3 sets. Tri being 3, somy referring to the number of sets of chromosomes. You've probably heard about trisomy before because you've heard about Down syndrome before. Down syndrome is trisomy of chromosome 21. So folks with Down syndrome have 3 copies of the 21st chromosome. And just that one, not of chromosomes 1 through 21, just of chromosome 21. If you have 1 set, we call it a monosomy. Mono being 1. One set of chromosomes. An example of this would be someone that only has 1 X chromosome. We call that Turner syndrome. So Turner syndrome, you did not get a second X chromosome from your parents, you only got 1, and so you have a monosomy. Pretty cool, right? Let's do a little bit of review. We started identifying what mutations were. So mutations are permanent changes in the DNA sequence. They're not always bad, even though we kind of talk about them like they're bad. They're not always bad. We can get a mutation either by inheriting it from our parents, and if we do inherit it from our parents, we call it a germline mutation, and it's present in all of our cells. It's present in all of our cells because it was found in the sperm or the egg of our parents. And our whole bodies are made from mitosis from our zygote, which is the fusion of the sperm and egg. The other way you can get a mutation is if you acquire it after birth. If you acquire it after birth, it's not in all of your cells, it's in some of your cells. And it's usually caused by environmental factors. An example would be, inhaling pollution, eating toxins, being a smoker. If I was a smoker, my mutation would occur in my lungs and upper respiratory tract. So it's in some of my cells, but not all of my cells. We have 3 types of mutations. We have mutations that don't change the sequence of a protein. And that's because even though your codon is changed, your overall amino acid is not changed, and so your protein doesn't change. So these are called silent mutations because they don't really have an effect on our proteins. The next type of mutation is a missense mutation. A missense mutation changes your codon just like a silent mutation, but in this case it does change the amino acid. So you're changing an amino acid which will have an effect on your protein. The effect could be little or it could be large. It depends on the type of protein. The last type of mutation is a nonsense mutation. A nonsense mutation inserts a stop signal so your protein doesn't grow to its full length, and that usually kind of just destroys your protein. We call that a truncated protein. It's too short. It's usually not functional. Now, these permanent changes in our DNA, these mutations, can happen in 4 ways. It can either be an insertion of genetic information, so you're adding an extra nucleotide or you're adding an extra section of a gene or a chromosome. A deletion of genetic information, you're removing genetic information. A duplication, so you're repeating just a little bit of what's already there. A repeat expansion, in which you're repeating a lot of what's already there, several times. And a frameshift, in which the change can be an insertion or deletion or duplication, but the thing about a frameshift is it changes the whole DNA sequence from the point of the mutation onward. So it changes your whole reading frame, because we read our sentence, hence called a frameshift. We also have mutations where you can gain or lose a copy of a chromosome, and those are aneuploidies. If you gain one, humans usually have 2, you'll end up with 3, and we call it trisomy. If you lose one, humans usually have 2, you lose one, so you're left with 1, we call that monosomy. All right. Well, thank you for taking notes with me. I hope that helped clarify this chapter and organize it a bit in your mind. And as always, send me emails, ask me questions. I love to communicate with you all, so please don't hesitate to reach out. And I will talk to you soon. Have a great rest of your day.